very much an open question. And so one way you can see how far we are away from a good understanding of this is in a very simple experiment. There are round, so planaria are these flatworms that regenerate. When you cut them into pieces, every piece builds whatever is missing and they regenerate. Okay, that's planaria. You can cut them into pieces and they every piece regenerates to a normal planarian. So there are species of planaria that have round heads, and those cells are really good at building a round head and then stopping. So they stop when a round head is complete. Okay. Then you got another species of planaria that has a very pointy head, kind of a triangular head. And those cells are very good at making a triangular head. When you cut it off, it makes a triangular head and then it stops. So now I have a simple question. If I take a bunch of the cells from the round-headed guy and I stick them into the body of the triangular guy and I let them sort of get comfortable and sit around for a little while, then I cut the head off. What head shape are we going to have? Are we going to have, is one of the head shapes dominant to the other? Are we going to have an intermediate shape? Or are we going to have a planarian that never stops regenerating because neither set of cells is ever happy about the shape? That stop condition is never set. Okay, so now look, the important thing is not the answer. The important thing is, despite all of the papers in Nature and Science about the molecular pathways of control of stem cells and planaria and all these kinds of high-resolution transcriptomics and all this stuff, there is not a single model in the field that makes a prediction on this experiment. Why? Because every piece of data out there now addresses the hardware that enables individual cells to do cell things. Fine. But we have no understanding of what happens when the cells join together into a larger scale self that makes large scale decisions about head shape, head number, things like that in morphospace, that navigates morphospace by making these large scale decisions. We have absolutely no idea how those algorithms work. And the fact that we know how the stem cells work and have lots of molecular biology about that really hasn't affected this other question much at all. We just have no models for this.
Because it's too computationally complex, or for some other in-principle reason?
I don't think it's, I don't think it's because it's too complex, although it is too complex to calculate out directly. I think it's because conceptually we haven't found the right way to think about how cellular collectives make decisions. This is a collective intelligence problem. This is not a molecular biology problem. We've been thinking about this as a molecular biology problem. That's not what this is. This is trying to read the mind of a collective intelligence. Now, people think of collective intelligences as exotic things like anthills and bee colonies and things like this. These are collective intelligences. I want to remind everybody that we are all collective intelligences. We are all bags of cells. There is no cognitive agent that is like this single diamond that's not made of parts. That's sort of unchanging. We're all made of parts. Any cognitive agent is made of parts. And so your goal is to ask, how do those parts bind together to make decisions as a collective? Individual cells don't know what a head is. They don't know what round means. They don't know what triangular means, right? But the collective sure does. And so the collective is able to navigate morphospace in this way that we don't understand the algorithm. So if we don't even know how to think about this. Okay, so this is very isomorphic to problems in neuroscience, to problems in artificial intelligence. It's trying to understand the scaling of minds, and in trying to do that, we pose the following problem. Okay, standard tadpoles make, standard frog eggs make frog embryos, and everybody's kind of used to that, even though, you can I always remind my students, did you realize that if I gave you the frog genome, you couldn't tell me what a frog looked like, right? You could compare it to other genomes that you did know what, but that's cheating. Okay, so we asked the simple question: where does the goal of making a frog or tadpole really come from? And how hardwired is that? So what we did was we took some skin that we scraped off of an early frog embryo. We set it aside in a different environment, and we said, okay, now you're free to sort of reboot your multicellularity. You're here. We've relieved all of the constraints of the rest of the embryo. You're no longer getting instructive signals from endoderm and from mesoderm and from all these other things. You're no longer subject to all these other signals. What are you going to do? What's your new, what do you want to do? And there's a couple of different options that could have happened. The cells could have died. They could have wandered off and sort of went each cell go its own way. They could have made a monolayer of cells in a dish, the way you get in cell culture. All kinds of things they could have done. They didn't do any of that. What they did instead was to combine together and to form a little ball that grew cilia, these little motile hairs on the outer surface. Now, cilia are normally sitting on the outside of embryos, and they're there to kind of redistribute the mucus around and to, make the pathogens sort of keep moving and not stick to the skin. They're used to keep the surface of the tadpole clean. But instead, these cells basically repurposed that genetically encoded hardware. The cilia themselves are genetically encoded. All the proteins necessary to make a cilium are in the genome, so they have that already. But what they did was they assembled themselves into a new kind of architecture, this spherical thing, and then they used the cilia to propel themselves. So they started running around. They started moving around, and so we have these amazing videos of them moving around singly, moving around in groups, interacting, going through a maze, going back and forth in various configurations. They have all kinds of behaviors. They have all kinds. They can regenerate. If you cut them almost entirely in half, they will like join back and make a make a xenobot again. And so the coolest thing about them is. And by the way, we don't know their cognitive capacities yet. Okay, we're only beginning now to start to see, can they learn? Do they have preferences? All these kinds of things. We don't know yet. But the coolest thing about them is that, to my knowledge, they're the only creature on the planet that doesn't really have an evolutionary backstory. The individual cells do. Okay, the cells have a long evolutionary history on Earth. But they were selected for, that genome was selected for the ability of these cells to sit quietly on the outside of the frog and keep out the pathogens. They were not selected specifically to be able to get together and run around in a separate configuration away from the embryo. Where did that all come from? And in fact, one of the things about it is people often say, well, when are you going to engineer these things? Knock in various synthetic biology circuits, right? Make them do things. And we absolutely will do that. But my goal, before we do any of that, my goal was to show people what can happen, all the diversity that can happen from the exact same genome with no manipulations whatsoever. Because the thing about these xenobots is they just have a normal frog genome. They have no transgenes, no genomic editing. There's nothing different about them. What we're seeing is the plasticity of this collective intelligence that is able to make a new functional proto-organism in a novel way out of the exact same parts.